So the topic of today's lecture is research and development and registration of drugs. And the drugs started to appear when the people injected the drugs and started to record the uh, progress of that, and it was from ancient times. And nowadays the drug development is a business that is very profitable, and before the drug can be used and reach the market, there are some requirements to pass, and uh, that takes a very long time. And usually the uh, usually the most common first step in the development of a new drug uh, is a discovery of a potential compound, or uh, that can be of the biologic uh, properties, or the can be synthesized chemically, and so these are two sources for the lead compound. And usually this compound has a new drug target. When a new drug is synthesized or discovered, the next step is uh, understanding its interactions with biological targets. Repeated application of these leads to compounds with increased efficacy, selectivity, and uh, mechanism. So, and usually that is done on animal testing. So, actually the first step and second step takes 2 years each. So, that first two steps are done in 4 years average. And the safety and efficacy of drugs must be defined before it can be sold out on the market. And also in vitro studies, relevant biologic uh, uh, profiles, and drug metabolism, pharmacokinetic profiles, and safety of the drug must be studied in animals before human trials can be started. And then after approval on and animal trials, so the first phase, or the first phases of clinical trials can be started. So these studies are on humans. Uh, and the fourth phase is actually is safety monitoring, and uh, is done after marketing is started. So And after approval, the drug can be dispensed by any uh, pharmacist, and also prescribed by any physician. So the analysis indicates that uh, modern new drugs uh, are actually not new. So most of the true original drugs are developed in public institutions like universities and research centers, and because the uh, the to develop the drug, the investment is required, and also it is required to integrate multiple technologies. Nowadays, most new drugs are developed in pharmaceutical companies, and the cost of the development usually is several hundred millions USD, and can be up to 1 billion or several billions. And so that is for single drug. And not all drugs will return the investment. It uh, only 20% of the drugs will return the research and development investment, and these are usually the blockbuster drugs. And to develop a new drug, usually the research institute will synthesize and test thousands of the compounds, and after the discovery, this compound will need to be optimized for potency, selectivity, and metabolism, and also for the dosing. And even after approval, there are some tests and that can increase the costs of the new drug development. And nowadays, only a few amounts of new drugs uh, are uh, can significantly increase uh, the uh, the usefulness of the drugs, so and are useful in the treatment. And more and more drugs uh, are only the analogous of the existing drugs uh, that are very popular. So and a similar drug can be developed to bypass uh, the marketing, uh, to bypass the uh, path, patent, so and also to get a profit. Uh, and despite the uh, large investment for the development, the drug market is most expansively growing, and most ex uh, most expensive one in the world. So, and also it is the most expensive business uh, and industry. So um, and the global investment for the pharmaceutical industry uh, nowadays is about several trillions of United States dollar. So uh, and this leads to the high prices of the drugs. So uh, when the new drug is discovered, the cost of the drug is very high, uh, and companies try to hold it very high because they need to get as much profit as possible to cover the cost of the research and development. And then later the price can drop down. So, and the companies have to develop a new drug to get a large return, to get a profit. So most new drugs are discovered through the following approaches. First is identification or elucidation of a new drug target. So this is actually the in vitro studies. Second is rational design of a new molecule based on an understanding of biological mechanism and drug receptor structure. Third is screening for biologic activity of large numbers of natural products, banks of previously discovered chemical entities, or large libraries of peptides, nucleic acids, and other organic molecules. The fourth is chemical modification of a known active molecule, resulting in uh, an analog of the drug. So this is so-called me-too drugs. So usually first two steps are carried out in universities and research labs. So these mostly academic institutions. So our university is one of them. But the cost of steps 3 and 4 usually can only be carried out in industry because it's very expensive and the universities usually don't have enough uh, uh, budget for these steps. Once a new drug target or promising molecule has been identified, the process of moving from the basic science lab to the clinic will begin. This translational research involves the preclinical and clinical steps described next. So, regardless of the source or the key idea leading to a drug candidate molecule, testing it involves a sequence of experimentation and characterization called drug screening. Uh, a variety of assays at the molecular, cellular, organ system, and whole animal levels are used to define the activity and selectivity of the drug. The type and number of initial screening tests depend on the pharmacologic and therapeutic goal. For example, anti-infective drug may be tested against a variety of infectious organisms, some of which are resistant to standard agents. Hypoglycemic drugs may be tested for their ability to lower blood sugar. Antihypertensive drug will be tested to lower the blood pressure, and antiarrhythmic drug to lower the arrhythmia, etc., etc. So the molecule will also be studied for the broad array of other actions to determine the mechanism of action and selectivity of the drug. This can reveal both expected and unexpected toxic effects. Occasionally, an unexpected therapeutic action is serendipitously discovered by a careful observer. For example, the antiviral agent amantadine was uh, was developed as a drug for the heart disease, but eventually during the sorry, it was developed as uh, anti-Parkinsonian drug. So, but antiviral properties were discovered unexpectedly. Uh, the selection of compounds for development is most efficiently conducted in animal models of human disease, where good predictive preclinical models exist. Uh, so, good drugs or breakthrough improvements are conspicuously lacking and slow for diseases for which preclinical models are poor or not yet available. For example, the Alzheimer's disease and autism. And studies are performed during drug screening to define the pharmacologic profile of the drug uh, at the molecular, cellular, organ system, and organism level. The value of this test is highly dependent on the reproducibility and reliability of the assays. For example, a broad range of tests would be performed on a drug designed to act as an antagonist for a neurovascular target before the treatment of hypertension. At the molecular level, the compound will be screened for activity on the target, for example, receptor binding affinity to cell membranes containing the homologous animal receptors. Early studies would be done to predict effects that might later cause undesired drug metabolism or toxicological complications. For example, studies on liver cytochrome P450 enzyme would be performed to determine whether the molecule of interest is likely to be a substrate or inhibitor of these enzymes or to interfere with the metabolism of other drugs. Effects on cardiac ion channels such as the ERG, potassium channel, or the possibility predictive of life-threatening arrhythmias are considered. Effects on the cell function determine whether the drugs is a agonist, partial agonist, inverse agonist, or antagonist at the relevant receptors. Isolated tissues, especially vascular smooth muscle, would be used to characterize the pharmacologic activity and selectivity of the new compound in comparison with reference compounds. Comparison with other drugs would be also undertaken in other in vitro preparations such as gastrointestinal and bronchial smooth muscles. At each step in this process, the compound would have to meet specific performance and selectivity criteria to carry it further. Whole animal studies are generally necessary to determine the effect of the drug on organ systems and disease models. Cardiovascular and renal function studies of new drugs are generally first performed in normal animals. Studies on disease models, if available, are then performed. For a candidate antihypertensive drug, animals with hypertension will be treated to see whether blood pressure was lowered in a dose-related manner and to characterize the effects of the compound. Evidence will be collected on duration of action and efficacy after oral and parenteral administration. If the agent possessed useful activity, it will be further studied for possible adverse effects on other major organs, including the respiratory, gastrointestinal, endocrine, and central nervous system. These studies might suggest the need for further clinical modification, compound optimization, and achieve more desirable pharmacokinetic or pharmacodynamic properties. For example, oral administration studies might show that the drug was poorly absorbed or rapidly metabolized in the liver. Modification to improve bioavailability might be indicated. If the drug was to be administered long term, an assessment of tolerance development would be made. For drugs related to or have a mechanism of action similar to those shown to cause physical or psychological dependence, abuse potential would also be studied. Drug interactions would be examined, and the desired results of this screening procedure uh, is a lead compound. It as a leading candidate for a successful new drug. A patent application would be filed for a novel compound that is efficacious or for a new and non-obvious therapeutic use for a previously known chemical entity. So uh, the first step is the actual preclinical safety and toxicity testing, and all drugs are toxic at some dose, and seeking to correctly define limiting toxicities of drugs and the therapeutic index comparing benefits and risks of new drugs is an essential part of the new drug development process. Most drug candidates fail to reach the market, but the art of drug development consists of effective assessment of management of risk versus benefit, and not total risk avoidance. Candidate drug must survive the initial screening procedures, must be carefully evaluated for potential risks before and during clinical testing. Depending on the purpose used of the drug, preclinical toxicity testing includes most of the procedures, and that includes the acute toxicity, subacute or subchronic toxicity. Chronic toxicity effect on reproductive performance, that is for pregnant women, and carcinogenic potential and mutagenic potential is tested. So I think that is all for today, and see you next week.
Okay, thank you, sir. Have a nice day.
Have a nice day too. Bye bye.
Goodbye.